that there's an incentive to renew the materials we're using into new things and continue the cycle. So in this particular case, my main focus is on taking polyethylene terephthalate, or number 1 plastic, and finding ways to degrade that, turning it back into its starting compounds, so that can either be used again for PET plastic without loss of quality or properties that would be ideal for consumer goods, or turning it into something totally new. So instead of having no outlet for that to go to, that's kind of the goal. And we study several other things, including PET, but kind of in this context, we're part of a large consortium called Bottle. It's bio-optimized technologies to keep thermoplastics out of landfills in the environment, which is a bit of a mouthful. So instead, the name Bottle is a lot easier to go with. And so our group is also part of that consortium.
And that consortium is formed by which kind of stakeholders in particular? Who would you be working with in that?
Yeah, so it's a big team of researchers at both industry, national labs, so government lab level, and academic labs across the United States, and then also there are some partners overseas. So we certainly are also involved with some researchers in the UK, and the partnerships and network are constantly growing. So the consortium just kicked off in the middle of last year. There was obviously some groundwork in place before that, but the team is ever growing.
That's great. And I mean, you kind of answered the first question I had, but I will repeat it anyway, just so we have it. What problem are you and your team solving? Now, you've obviously just highlighted that. And then, so perhaps we can move on to, please tell us a little bit about the research and you've been doing and the importance it has for closing the loop of plastic production.
Right. So it might be easy to give just a little bit of background on PET so that this is in some context. I won't claim to be an expert on all of the steps in this process, but so PET is one of the most highly produced synthetic polymers that we use in consumer goods and industry and packaging, and also in industry and textiles. So it's a lot of markets to cover, and it's just extremely versatile and easy to produce. The starting materials all come from petroleum feedstocks. So they're refined petroleum feedstocks, but the building blocks are terephthalic acid and ethylene glycol. So if you squint at a PET chain, which is kind of a cool, so polymers are all these small monomers that come together to form long chains. If you squint at it, it could almost look like some of the natural polymers that we find in the environment. So the research that we're doing is looking at biological solutions to breaking down this synthetic polymer and either recreating the starting components or turning those into something totally new. And so my specialty is in biochemistry, so I'm mostly focused on the enzymes that would do this. So enzymes are protein machines that they do everything that you can imagine in biology. In your body, you have gazillions that are doing all of the functions that you would need to survive. And in every living organism, there's that similar systems. But in this case, we're mostly looking right now for enzymes that have some specificity or affinity for these types of synthetic polymers, and they can break the bonds that form that chain, releasing these small components. So either those are small chunks or they're back to the monomer that started. So instead of needing to draw from that petroleum feedstock over and over again, you could use the material that was created initially and regenerate the same material without loss of quality. So I keep going back to this loss of quality concept because PET, while it is the most abundantly produced synthetic polymer right now, is also the most recycled polymer right now. So if you think about curbside recycling in some of the countries in the world, this is the easiest to handle, and also has probably the largest financial impact. So if you can collect pretty pristine and clean drinking bottles, for example, many of those can be recycled mechanically. The problem with mechanical recycling is that it involves crushing the material and shredding it, melting it down and turning it back into a new product. But the new products are of lower quality. They don't have the same mechanical properties or thermal properties that the original material did. So you're downgrading the material over time. And I think statistically, when you look at the large system, materials might get recycled once, maybe less than that, and only about 20% of the materials that are made in this polymer class ever actually enter a recycling stream. And so we have a large resource that we could tap into and approach this in a different way, potentially a way that would result in even more recovery of the original material, meaning that you have to dip into other resources even less.
And it's great, and we really appreciate and sort of find we're looking very much at how nature can come into these equations, because obviously it's been kind of kept out of the equation, if you like, for 80 years, and now this research you're kind of looking at is very much drawing on how natural products would or natural sort of environments would work, and we're interested in that, how in particular you perhaps came across this discovery by mimicking nature or investigating how nature was kind of building up around some of these recycling plants, which is to understand how this developed. But could you tell us a little bit about how the team came to this kind of discovery and how it started looking at the particular kind of biochemist approach to plastic?
Sure. So I'm going to start in the middle of the biggest story, because this field has been in existence for a few years longer than our team has been working on it. But in 2016, a group from Japan identified and published a story that made a really big splash in both the academic literature, but also the public media, that they had identified an organism growing in a recycling facility in Japan, and it can use as its sole carbon source for this organism PET bottles. So it was somehow living on these old plastic piles, and it didn't have any other food source. So if you think about that, it's really remarkable, because it means that this organism found itself in a place short of other nutrients to eat, and it evolved over a really quick time to be able to take in a synthetic polymer. And if you think about all of our uses of plastic, the goal is that biology doesn't break them down. Right? All of the materials that we have developed, they're incredibly useful, in large part because they don't break down, that we can use them over and over again, or they have a lot of durability and resistance to all of the things that we might do to them. But that of course encompasses all plastics, and for certain applications that's not necessary. So in this case, we have a material that's really resistant to biological activity, and instead this organism found a way to do that anyway. And I don't want to confuse anybody that it only wants to eat PET. If there were other food sources around, this organism would certainly like to eat those instead. But in the lack of other dinner, it made something worthwhile, right? So the way that it did this was to probably, this is hypothetical, but likely what happened is that this organism evolved from a different type of food source that looked similar, had similar chemical linkages, and it co-opted those same proteins that would break down those natural polymers to be really specific for this synthetic polymer. And the two enzymes that do that are PETase and mHETase. So in biochemistry, we add the ending ase onto the name to indicate that it's an enzyme that breaks down something. So PET is the PET plastic. So we have the first enzyme that breaks down PET plastic, and then the second enzyme in the pathway breaks down small monomeric chunks that come off of that in the breakdown product. So the final resulting compounds are terephthalic acid and ethylene glycol, which is what you started with to create the material. And then this organism can use those as food. So it has the catabolic pathways to eat terephthalic acid and ethylene glycol. So it's not exactly eating PET, it's doing something to it to crush it up and make it into edible little morsels. But that's really impressive, especially when thinking on the grand scale for biology, because PET wasn't invented until the 40s. So 1940 to 2016 isn't a very long time on the evolutionary time scale to think about a new adaptation. And so it was able to do this quickly, possibly because the materials look similar enough that it could easily adapt. But that also means that it's not perfect. It's not a really fast process, it's a slow process, which means that these organisms don't get a lot to eat. They have to work pretty hard for their meal. So the reason our group got interested in this is that this isn't the only enzyme that's ever been reported to work on PET plastics. There's several others. So that's why I'm saying I'm starting in the middle. But our group got interested in this because this one is very specific to PET. And so we wanted to understand what might be the structural differences or in the protein sequence that might give it this capability to be really specific for this polymer. So the field in general, probably we could give its origins to more like the early 2010s, 2010, 2012, that sort of area, where these other enzymes were identified that are specific to cutin and suberin. So these are materials that are part of plant mass. And they are, like cutin is the waxy layer that covers leaves and gives sort of some water and water impermeability to plants, so that invaders find it more challenging to get into the plant, and also it keeps the water balance at equilibrium. So that's cutin is part of that process. So the waxy cuticle layer on a leaf, and suberin is much like cork. So in a cork tree or materials that feel like cork, you would find suberin, and these are complex natural polymers that look in some of their linkages a lot like synthetic polymers. So that's what it was found in those earlier studies, that there are some enzymes that exist that can already do this, but that they're not specific to PET. They just happen to do it, and some of them do it quite well. And so this is just building this body of literature and understanding of what might make an enzyme happy to break down a PET polymer, and how do you then, moving forward, how do we then make that better? How could we encourage that process to be faster or more durable, or to work on more and more types of PET that come in different from different types of processes that created the material to begin with. And so this is where the research is going.
Which is where the double mutant comes in, correct? I mean, there was this breakthrough recently where it's discovered that obviously that there were certain enzymes that work better than others, and just like playing around with the bacteria that they discovered there was also a way to accelerate the process as well. But how has that research progressed since the discovery was released in 2018?
Right. So our group got interested in PETase because of that specificity question, that it's evolved specifically for PET. And so in collaboration with the John McKeon's group at the University of Portsmouth, who are wizards at structural biology, and our group at NREL and several others, there's always a lot of collaboration in these projects. We explored what we could do with PETase, and then made some targeted mutations to see how that would change either specificity or activity. And what we found is that this double mutant, so it has 2-point mutation, so 2 amino acids in the whole sequence have been changed. And what this actually did was to make PETase look a little bit more like those cutinases that I mentioned that had been discovered previously. And what this did was allow an improvement in some cases for the reaction rate. So we found that even more degradation occurred when you use this. And that was awesome. And in terms of a first step, we're still talking about a really new field here. And the structure, having the structure in hand helps us to understand how an enzyme might work, and then what we might be able to do to make it work better. And that's also to give comparison between all of these different natural variants that would occur. So moving on from there, we then looked into the second enzyme in the pathway, M-headase. So this is also from this organism that was discovered. But the second enzyme it's strikingly similar to PETase, except that it has a full extra portion of the enzyme that makes kind of like a hat or a lid on top of the reaction site. And this enzyme is quite particular to the smaller components. So it still does a similar reaction where it will break an ester bond, but it is only going to work on this M-het compound. So we wanted to know if this could be optimized or changed, and looked at a lot of different mutants of this enzyme as well. But then, because they work together, and this is a system, a team, we also wanted to figure out how each of those enzymes was participating in this partnership, and would there be some circumstances in which that could be by using the two together, we could improve performance. So in 2020, we also published a study describing how this partnership is really important, and that activity, so the amount of PET breakdown that you can see with PETase alone is quite a bit less than when PETase and MHETase are present together in the same reaction. But to go a step further, just to see, we also tethered those together so that they would make a complex. And so this is a chimeric protein. And when tethered together, the performance is even better. And so this is all still really preliminary work, and all of our reactions are done at really small scale. But what this is doing is starting to show some of the details about how we could implement different strategies for using enzymes to create larger processes or larger scale industrially relevant processes for handling PET waste.
Yeah, I was going to ask, jumping some of the questions I had sent you earlier, but how can you kind of envisage then this biotechnology, the PETase and other ketones being applied to the problem the world faces of plastic pollution? And what are the real challenges applying these structures at scale? Have you identified the kind of a roadmap towards actually being able to apply these discoveries in the real world?
Yeah, so this is definitely still a work in progress, but as part of our group at NREL, we have techno-economic analysis teams. So they do analysis on all of the factors that you would need to consider in order to create a process at scale. So that includes, what would the feedstock need to look like? Where would that come from? What would it cost? How would you build a facility? What would be involved in that? What would the process look like? Where would the materials go at the end? And the idea of this is that not only can you kind of glean this data from other processes that exist and then make some assumptions along the way to build this for a totally new application, but it tells you where areas are really expensive to make into reality and to help guide what research still needs to happen to make this an economically viable process. So we have this techno-economic analysis underway. It should be available to the world soon. It's in the works. And what it shows, actually, there's some really impressive results coming out of this, in that there are circumstances in which we could get cost parity or really close to the same cost for an enzymatic recycling process of PET to the virgin production of PET. And part of that is not just the money side of it, which you need to have some sort of economic incentive for the world to adopt a process on the global scale. But there are a lot of other benefits to this process, and especially in terms of greenhouse gas emissions and energy consumption and water consumption that look really different along the supply chain. And so this is all work that's definitely been happening. And the results, at least at this preliminary stage, are extremely promising. That while we may not quite be to that stage where we can implement this, there are some groups in the world that are much closer than we are. The process itself could be economically viable. So the next steps are a lot about scale up and finding the appropriate feedstocks, and then finding appropriate buyers at the other side to make a new product from the materials.
Yeah, I mean, so this obviously there's a lot of energy going into this particular problem, from all the different stakeholders, whether it's scientific community, financial, the finance industry. Obviously, everybody's focused on this as one of the greatest opportunities and challenges, facing us as a species. How does your research make you feel perhaps optimistic about the problem? And I wanted to actually, before perhaps we talk about that, is talk a little about how we got to where we are in the first place. How science, kind of came forward with a solution without necessarily having the answer to the problem. Obviously, it's a bit harsh on people to perhaps kind of make them imagine what could have possibly happened in 1947 when they created this polymer, to imagine the final outcome. Could you, as sort of from your particular place, give us some context about how, when you're looking at creating new solutions to the world's problems, how you look at applying systems theory to this and making sure that whatever you do is kind of future-proofed or has at least some sort of thought process gone into the possible consequences from that. Because when we look at the history of PET, who would have thought that this product, but basically this kind of chemical solution, would have brought us to where we are right now? Obviously, with the confluence of many different people, many different actions, the human behaviour, all of the different things that happen to bring us to the problem where we now need a solution, which is what everybody's working to find on plastic pollution, obviously. So, can you, from the scientific point of view, give us a short, potted history of how we got to where we are right now, and and how that shapes your thinking moving forward? In terms of coming up with solutions that aren't going to make the problem worse, or that hopefully will bring systems back into balance.
Yeah, absolutely. So I guess it's hard to give a full overview from only a scientific perspective, as I'm also, this also affects my life personally. But starting in the 1940s, where obviously it was very hard to envision how the world would change over time. But with the advent of these quick, cheap, and safe materials to package food and to use for all of the applications you can think of, every piece of clothing and device that you own has probably some plastic in it, including this phone that I'm holding right now for recording this. It impacts every aspect of our life. And this isn't just for convenience, but also for health and safety. So medical devices, the medical field has been able to accelerate its ability to treat disease and to, with sterility, move through all sorts of things that had been deadly for centuries that are now not even thought to be a problem, really, a quick solution. And all of this, it's revolutionized the way that we do almost all of the activities in our life. And in more developed countries, this is, I think, taken a bit for granted because it's so pervasive. But this is extremely important in the developing world as well, where the materials that have been replaced, they make a big difference to the quality of people's lives, the sanitation aspects, everything. And so we are in a sticky place at this point with the accumulation of these materials that we've made. They don't go away the way that natural materials do. And part of that is because there aren't very many organisms that have evolved to be able to handle them. And in some ways you might look at that as just being a matter of time, but we're not at a point where we can handle the inputs to continue to increase when the outputs don't ever disappear. And so while that is, I guess, a bad situation to be in, I don't want to demonize plastic as the worst thing that's ever been created. There are so many societal and health and technological impacts that it has had that have been crucial in the development of the globe at this point. And so I think a lot of that opportunity and possibility was what the initial creators were envisioning all of those decades ago.